All right, let's dive into DNA. You sent over a ton of research, so we're going to break down why it's so important to life. Looks like excerpts from a chapter on DNA structure and organization, even some cool experiments.
Yeah, it's a wild ride, the history of figuring this stuff out.
Totally. So you've been in the DNA research. What's got you hooked on it being the blueprint of life?
Well, it's DNA, deoxyribonucleic acid, right? It's got the instructions for like everything about a living thing, how it
Hmm.
develops, how it works, how it grows, even how it makes more of itself. It's all in the code.
So like a super detailed manual, not just for building, but for actually running the whole living thing.
Exactly. And not just any molecule can pull this off. There are requirements to be the carrier of genetic information.
Hmm.
You know, got to pass the test.
Okay, so what's on this molecular superstar checklist?
Well, first off, it's got to copy itself perfectly every time, right? So when a cell divides, the info gets passed on without any mistakes.
Makes sense. So perfect replication, that's one. What else?
It's also got to hold a massive amount of information, like think billions of lines of code.
Okay, a blueprint that's huge. So storage is important. What else?
Well, you got to be able to use the info, right? So it's got to be able to express that information, meaning transloading the code into actual traits.
Yeah.
What makes you you.
And finally, you got to have a little wiggle room for changes, right? Those little mutations that allow species to evolve and adapt.
Exactly. Got to be able to change with the times.
Uh,
And DNA does all this through a really neat process. Basically, DNA gets transcribed into RNA, and then RNA is translated into proteins. Central dogma of molecular genetics, it's called. DNA to RNA to protein.
DNA to RNA to protein, got it. But wait, didn't scientists used to think proteins were the big players in heredity? Not DNA.
Yeah, you're right. Science changes what it thinks sometimes.
So why proteins? What made them seem like a better bet for a while?
Well, back then, scientists knew way more about protein chemistry than DNA. They seem, more complicated. They've got 20 amino acids as building blocks, and DNA only has 4 nucleotide bases. Seems simpler. Plus, they could actually see all the different jobs proteins were doing in the cell.
So DNA at the time just seemed too basic to be carrying all that complex genetic information.
Yeah, there was even this theory, the tetranucleotide hypothesis. Came from Phoebus A. Levane. He thought DNA was just this boring, repetitive molecule with, like, equal amounts of the 4 nucleotides, not nearly complex enough to be in charge of all the instructions.
Wow, talk about a scientific plot twist. What changed everything?
Well, a bunch of experiments totally changed the game, showed just how important DNA really is.
Okay, gotta hear about these game changers. Which ones really stand out as proving DNA's the boss?
3 big ones really shifted things. Start with Griffith's transformation experiment, 1927. He was working with 2 types of bacteria, Diplococcus pneumonia. One was like the pneumonia-causing kind, and the other was harmless.
How could he tell them apart?
So in the lob, a virulent strain, the bad guy, it formed these smooth, shiny colonies. They called them S. The harmless ones were rough. They called those R.
Smooth equals danger, rough equals safe. Got it. What do you do with these bacteria?
Injected them into mice. So inject the smooth strain, mice die. Inject the rough strain, mice live. But here's where it gets wild. He heat-killed the smooth strain, made it harmless, then he mixed that with the rough strain and injected them together.
Okay, 2 harmless strains together. What happened?
The mice died. And get this, when he looked at their blood, it was full of living smooth bacteria. Like something from that heat-killed smooth bacteria had transformed the rough bacteria, made them deadly.
No way. Did he know what caused that transformation?
He called it the transforming principle, but he had no idea what it actually was. Took another 17 years to figure that out.
All right, you got me on the edge of my seat. Which experiment cracked the code?
The Aver-McLeod-McCarty experiment, 1944. They continued Griffith's work. Same smooth and rough strains, but they went a step further.
What did they do differently?
They took the heat-killed smooth bacteria extract and, removed stuff one by one. Took out the proteins, the carbohydrates, the lipids. Each time, they tested if it could still transform the rough bacteria.
So like a molecular detective game trying to isolate the transforming principle. What'd they find?
They got rid of everything they could think of, but the transformation still happened. But then they used an enzyme that breaks down DNA, and boom, no more transforming ability.
So it was a DNA all along. No magic, just science.
Exactly. First real proof that DNA, not protein, was the star of the show. Of course, some scientists were still skeptical. It was a big deal.
Big claims need big proof.
Totally. You need lots of evidence. And that's where the Hershey-Chase experiment comes in, 1952. They used bacteriophages, those viruses that infect bacteria.
Bacteriophages sounds kind of like tiny pirates infecting and taking over.
Perfect analogy. They inject their genetic material into bacteria, hijack the cell to make more viruses.
Mm.
Hershey and Chase wanted to know, what's doing the hijacking? Is it the protein code of the virus or the DNA inside?
Clever. How did they figure that out?
Radioactive isotopes, like little tracking devices, labeled the protein code of some phages with radioactive sulfur and the DNA of others with radioactive phosphorus.
Two sets of glowing viruses, one with glowing protein, one with glowing DNA. Then what?
They let these labeled phages infect bacteria. After a bit, they used, get this, a blender. Detached the phages from the bacteria without breaking the bacteria open. Then they used a centrifuge, separated the bacteria from the loose phage parts.
So the radioactive signal would show them where the genetic material ended up.
Right. And guess what? The radioactive phosphorus, the DNA label, it was inside the bacteria. The radioactive sulfur, the protein label, outside. Meaning it was the DNA that got injected and gave the instructions for making new phages.
Case closed. 3 different experiments, all pointing to DNA as the hereditary hero. What a team effort. So what's the next chapter in the story?
Well, they knew DNA was the key, but now they had to figure out how it actually worked. That kicked off a race to decode the structure of DNA itself.
I can imagine scientists scrambling to figure it out. What was that like?
So picture it. 1950s. Scientists around the world are trying to crack the code. How do these 4 bases, adenine, thymine, guanine, cytosines, how do they fit together to make this molecule that holds so much information?
Did they have any clues?
They did. 2 big ones helped them solve the puzzle. The first clue came from a guy named Erwin Chargaff. He was looking at DNA from different organisms and noticed something interesting.
What'd he find?
He found that the amount of adenine was always the same as the amount of thymine, and the amount of guanine was always the same as cytosine, every time.
So, like a balance, A and T, G and C, always paired up.
Exactly. We call them Chargaff's rules now, and they were a big hint that these bases were somehow connected.
That makes sense. What was the other clue?
X-ray diffraction images of DNA.
X-ray diffraction. Remind me how that works.
You shoot X-rays at a crystal of a molecule. The X-rays scatter in a pattern, and that pattern tells you how the atoms are arranged. Like a shadow picture of a molecule shows you its shape.
Got it. Did they get a good picture of DNA with that?
Oh, yeah. Thanks to Rosalind Franklin, she was amazing with X-ray diffraction. She got these really clear images of DNA while she was working in Maurice Wilkins' lab. Her work was crucial.
What did her images show about DNA?
They showed this X-shaped pattern, like a clue that DNA had a helical structure, kind of like a spiral staircase.
Okay, so we've got Chargaff's rules hinting at base pairing and Franklin's X-ray images saying, hey, it's a helix. How did it all come together?
Enter James Watson and Francis Crick, 2 brilliant guys, totally obsessed with figuring out DNA structure. They took all the evidence, Chargaff's rules, Franklin's X-ray data, and built models, tried to make it all fit.
This is it, the big reveal. What did they figure out?
1953, they published their paper, proposed the famous double helix model of DNA.
The double helix, one of the most famous images in science. What does it actually look like?
Imagine a ladder, twisted. The sides of the ladder are made of sugar and phosphate molecules. That's the backbone.
Okay, I'm picturing it. What about the rungs?
Here's the cool part. Each rung is a pair of those bases, either adenine with thymine or guanine with cytosine, just like Chargaff's rules predicted.
A with T and G with C, it all connects.
Yep. And these pairs are held together by these weak bonds, hydrogen bonds. They keep the 2 strands together but can also come apart when they need to, like when DNA replicates.
What else is important about the structure?
The 2 strands run in opposite directions. It's called antiparallel.
Antiparallel, what does that mean?
Think of it like this. One strand runs from, we call it the 5' end, to the 3' end. The other runs from the 3' end to the 5' end. Those primes refer to specific carbon atoms in the sugar molecule. And this opposite arrangement is super important for how DNA works.
So elegant, the double helix. Explains how DNA can store info, copy itself, even change with mutations.
You got it. This discovery changed everything, revolutionized biology. It's like one of the biggest scientific breakthroughs ever.
So we talked a lot about DNA. What about its cousin, RNA? How is it different?
Right? RNA, or ribonucleic acid, it's like DNA's close relative, lots of similarities, but with some key differences.
Okay, hit me with the family drama. What's different?
First, the sugar's a bit different. RNA uses ribose instead of deoxyribose, hence the name ribonucleic acid.
Different sugar, check. What else?
And instead of thymine, RNA uses a base called uracil. So in RNA, you've got adenine pairing with uracil, but guanine still pairs with cytosine.
Interesting. So slightly different base pairs. What about the overall structure? Is RNA also a double helix?
Nope, that's another difference. DNA is usually a double helix. RNA is mostly single-stranded.
Single-stranded, so no base pairing at all.
Not necessarily. RNA can fold back on itself and make double-stranded sections, and those are important for how it works.
So it's a bit more flexible than DNA.
Exactly. And that lets RNA do all sorts of things in the cell.
Speaking of doing things, what are the main jobs of RNA?
Okay, so there are three main types, each with its own gig. First, ribosomal RNA, or rRNA. It's a key part of ribosomes.
Ribosomes, those are the protein factories, right?
You got it. They're where the code from DNA gets turned into proteins. And RNA helps build the ribosome and plays a role in actually making those proteins.
So RNA is essential for building proteins. What about the other types?
Next up, messenger RNA, or mRNA. It's like the messenger, taking genetic info from the DNA in the nucleus out to the ribosomes.
So like a blueprint delivery service taking orders from the main office to the factory floor.
Perfect analogy. DNA is like the master blueprint, locked up tight, and mRNA makes a copy of the part they need and delivers it to the ribosomes for production.
Got it. What about the third type?
That's transfer RNA, or tRNA.
What does tRNA do?
It's the translator. It brings the amino acids, the building blocks for proteins, to the ribosomes, matching them up to the instructions in the mRNA.
So tRNA is like a delivery service, matching the right amino acid to the right code on the mRNA.
Precisely. It's super specialized, with a structure that lets it recognize both the mRNA code and the matching amino acid.
Wow, it's amazing how these three types of RNA work together to make the proteins.
It's a beautiful system. It really shows how important RNA is in gene expression.
Sounds like there's a lot more to RNA than meets the eye.
Oh, yeah. More versatile than we thought. It even does things like gene regulation and telomere maintenance. But we'll have to save that for another deep dive.
I'm already looking forward to that one, but I think we covered a ton of ground just on this first part of our DNA exploration.
We did. From those early ideas about DNA being too simple to the elegant double helix, and even a glimpse into RNA's many talents.
Definitely. And all this from that one question, what is the molecule of heredity? Stick around, because in part 2, we're going deeper into how this amazing molecule actually works.
It's pretty wild to think, even after they figured out that double helix structure, scientists still had a ton to learn about how DNA actually did its job as the hereditary material.
Yeah, knowing what it looks like is one thing, but figuring out how it works, that's a whole other challenge. How does a double helix make copies of itself anyway?
Well, that's part of the beauty of it. The double helix, it's like a self-replicating blueprint.
Okay, not sure I'm following. What do you mean by self-replicating blueprint?
Remember those base pairing rules? A always with T, G always with C Char-Gas rules.
Right, the base pairing rules, they're important, yeah. But what do they have to do with replication?
They're the key to the whole thing. Because of that specific pairing, each strand of the DNA molecule can act like a template to build a brand new matching strand.
So you're saying if you pull those two strands of the double helix apart, each one has the instructions to make a new one.
Precisely. Imagine you have a mold, right, to make something. You can use each half of the mold to create a complete new object. It's the same idea with DNA.
That's pretty clever. But how does the cell actually pull the strands apart and build the new ones?
Enzymes. It's a whole team of specialized proteins, enzymes. Think of them like tiny molecular machines, each with its own little job to do.
So there are enzymes that can, like, unzip the DNA helix?
You bet. There's one called helicase. It unwinds the double helix, breaks those hydrogen bonds holding the two strands together.
Okay, helicase unzips it. What happens next?
Then another enzyme comes in, DNA polymerase. It's the builder.
DNA polymerase, the builder. Okay, what does it do?
It reads the sequence of bases on each of those separated strands, and then it adds the complementary nucleotides to make a new strand.
So if the template strand has an adenine, DNA polymerase adds a thymine to the new strand. And so on.
You got it. And it does this with incredible accuracy, very few errors, like a tiny, super precise copy machine for DNA.
Amazing. And the end result is two identical DNA molecules, each one with an old strand and a new strand.
That's right. But, before they figured out exactly how DNA replication worked, there were a few different ideas floating around.
Oh, really? What were some of the other theories?
Well, there were three main models for how DNA might replicate: conservative, semi-conservative, and dispersive.
Okay, break those down for me. What was the conservative model?
So the conservative model said that the original DNA molecule stayed together completely, and then a brand new double-stranded molecule will be created from scratch.
So like making a photocopy of a document, original stays the same, you get a separate copy.
Exactly. Now, the semi-conservative model is what we just talked about. The original DNA molecule splits, and each strand is used as a template to build a new one. So you end up with two molecules, each with one old and one new strand.
I see why it's called semi-conservative. Half of the original is conserved in each new one. What about that dispersive model?
That one was a little more complicated.
Aha.
It said the original DNA molecule would be broken up into pieces, and those pieces would be used to build new DNA molecules. So the new molecules would be a mix of old and new DNA, but not in a neat half and half way like semi-conservative, more jumbled.
A mixed-up blend of old and new. How did scientists figure out which one was actually happening?
More clever experiments. This time they used isotopes again, those radioactive tracers we talked about. In 1958, two scientists, Matthew Meselson and Franklin Stahl, they did this experiment that became super famous for proving the semi-conservative model. It's called the Meselson-Stahl experiment.
So what did they do in their experiment?
They started by growing bacteria in a medium that had a heavy isotope of nitrogen. It's called nitrogen-15.
So the bacteria would use that heavier nitrogen to build their DNA?
Yep. Then they moved the bacteria to a new medium with normal nitrogen, nitrogen-14.
And let them replicate their DNA.
Exactly. And after each round of replication, they took samples of the bacteria and used this technique called density gradient centrifugation to see how dense the DNA was.
So the density told them something about what the DNA was made of, right?
Right. The density depended on if the DNA had heavy nitrogen, normal nitrogen, or a mix of both. So they could track what happened to that original heavy nitrogen DNA during replication.
So they could see if the original DNA stayed intact, or if it was split in half, or even broken into fragments. What did they find?
The results were crystal clear. After one round of replication, all the DNA was halfway between the heavy DNA and the normal DNA in terms of density.
So that ruled out the conservative model right away.
Yep. If it was conservative, they would see two separate bands of DNA, one heavy and one normal.
Okay, so it had to be either semi-conservative or dispersive.
Right. But after two rounds of replication, they saw two distinct bands of DNA. One matched normal DNA density. The other was halfway between heavy and normal.
And that ruled out the dispersive model.
Exactly.
Yeah.
If it was dispersive, they would have kept seeing just one band getting less and less dense each time.
So the results matched the semi-conservative model perfectly.
Perfectly. The Meselson-Stahl experiment was a beautiful demonstration that DNA replication happens in a semi-conservative way, just like Watson and Crick had predicted.
It's amazing how scientists can use these tools to figure out what's happening at such a tiny level.
It really is. That experiment is a classic example of the scientific method in action. Observe, hypothesize, design an experiment, and get those clear results that either support or refute your hypothesis.
And in this case, the results beautifully supported the semi-conservative model.
They sure did. And understanding DNA replication this way has been huge for so many fields, medicine, biotechnology. It's the foundation of how we understand genetics.
Speaking of the double helix, the chapter you sent mentioned that the DNA molecule can actually twist and turn in different ways, even mentioned something called zDNA. What's that all about?
You're right. While that classic Watson-Crick model, that's a right-handed double helix. But DNA doesn't always have to exist that way.
Wait, there are other forms of DNA. What do they look like?
One is called zDNA, and it's a left-handed double helix.
So it twists in the opposite direction. Whoa.
Exactly. And it's got more of a zigzag shape, not that smooth curve of bDNA, the classic Watson-Crick one.
Why does it twist the other way? What makes it different?
Well, it comes down to how those sugar phosphate backbones are arranged and how the bases are stacked together. It's still a double helix, but with a different kind of twist.
Interesting. So the actual sequence of bases can change the shape of the DNA molecule. Is this zDNA common?
It's not as common as bDNA, but scientists have found it in certain parts of the genome, particularly in areas with lots of guanine and cytosine bases. We say areas with high GC content.
High GC content, meaning more G and C bases than A and T. Does zDNA have a specific function?
That's a great question. And honestly, scientists are still trying to figure that out completely.
Yeah.
We know the shape of the DNA molecule can affect how it interacts with proteins and other molecules. So it's possible that zDNA is involved in things like gene regulation or other cellular processes. We're still learning.
That's so cool. It's like there are hidden layers of information in the shape of the DNA.
It really is. There's so much we're still discovering about DNA. And it's not just the double helix itself that can twist and turn. The whole molecule can coil up on itself, too. It's called supercoiling.
Supercoiling. Sounds intense. What is that exactly?
Imagine you have a long piece of string and you start twisting it.
It's like twirling it between my fingers.
Exactly. As you twist it more and more, the string starts to coil up on itself, makes loops and knots.
I can picture that. But why would DNA need to supercoil? Wouldn't that make it more tangled?
It actually helps. Supercoiling makes the DNA molecule more compact so it can fit inside the cell. Remember, DNA is incredibly long.
Right. It's got to be packed in that tiny nucleus somehow.
Exactly. Supercoiling lets it all fit without becoming a tangled mess. And there are actually two types of supercoiling, positive and negative.
What's the difference?
Go back to that string we were twisting. If you keep twisting it in the same direction, it gets more and more coiled. That's positive supercoiling.
Okay. And if you twist it the other way?
We're basically untwisting it, right? That's negative supercoiling.
So which way you twist it matters?
It does. Because the level of supercoiling can affect how easily the enzymes can get to the DNA for things like replication and transcription, all those processes we talked about.
So it's not just about packing DNA efficiently, it's also about controlling access to it.
You got it. Supercoiling plays a big role in gene regulation, deciding which genes are turned on or off.
This is getting complex. The structure of DNA is connected to so many different functions.
Absolutely. We haven't even gotten to how DNA is organized into chromosomes, which are even bigger structures.
Okay, so let's zoom out a bit. What's the story with chromosomes? How does DNA get packaged into those?
So chromosomes are made up of DNA and proteins together. It's called a chromatin. And the basic unit of chromatin is something called a nucleosome.
Chromatin. That's the stuff that chromosomes are made of. Okay, and nucleosomes, what are those?
A nucleosome is like a tiny spool of DNA wrapped around a core of histone proteins.
Histones, those are proteins that bind to DNA, right?
Right. Histones have a positive charge, and DNA has a negative charge, so they're attracted to each other, like magnets.
So the DNA wraps itself around these histone proteins to form nucleosomes? Then what?
Those nucleosomes then get organized into even bigger structures, and eventually all that condenses down into those compact chromosomes that we can see during cell division.
It's amazing how it's packed in so tightly, but can still be accessed for replication, transcription, all of that.
It's a truly impressive feat of molecular organization. And the way it's packaged can actually influence which genes are expressed.
Wait, you mean the structure of the chromosomes can affect which genes are turned on or off?
Absolutely. If a gene is really tightly packed, it's harder for the cellular machinery to get to it to transcribe it. And if it's more loosely arranged, it's easier to access.
It's like having a library full of books. If the books are crammed on the shelves, you can't find what you need.
Perfect analogy.
Yeah.
And to make things even more dynamic, there are enzymes that can actually remodel the chromatin, make certain genes more or less accessible as needed.
So it's not just a static structure, it's a dynamic process.
Exactly. The way DNA is packaged into chromosomes is a major way that gene expression is regulated, fine-tuning which genes are active at any given time.
This is blowing my mind. We've gone from the tiny double helix to these huge structures, chromosomes. It's amazing how much information is packed into one cell.
It is, and we're still just scratching the surface of DNA's complexity. But there's one more super important concept we need to talk about. Mutation.
Right. Mutations, the chapter mentioned that. DNA has to be able to change, to vary through mutation. Why is that so important?
Because mutations, those changes in the DNA sequence, they're the driving force of evolution.
They introduce those new variations, right? Give natural selection something to work with.
Exactly. Those variations can be helpful, harmful, or neutral. Some might give an organism an edge in its environment, others might be bad for it.
So it's a bit of a gamble, but mutations are essential for species to adapt and change over time. What are some ways DNA can mutate?
One common one is a point mutation. That's where a single base in the DNA sequence is changed.
So instead of an A, maybe there's a C.
Yep. And those little changes can have all sorts of effects, depending on where they happen in the DNA sequence.
What kind of effects?
Well, sometimes a point mutation can change which amino acid a codon codes for.
Remind me, what's a codon again?
No, right, sorry. A codon is a group of 3 bases, and it tells the cell which amino acid to use. So if a point mutation changes a codon, it can change the amino acid sequence of a protein.
And that could change how the protein works.
Exactly. Sometimes those changes are good. Maybe the protein works better or does something new. But sometimes they can mess up the protein's function.
Like you said, a bit of a gamble.
It is. But mutations, they're the source of the variation that natural selection acts upon. Without mutations, there's no diversity, no evolution.
Makes sense. Are there other kinds of mutations besides those point mutations?
Oh, yeah. There are insertions and deletions, where you add or remove one or more bases from the DNA sequence.
So, like adding or deleting letters from a sentence.
Perfect analogy.
Yeah.
And when you do that, it can shift the reading frame of the DNA sequence.
Reading frame, what's that?
Remember, the DNA sequence is read in groups of 3, those codons. The reading frame is how those codons are read. So if you insert or delete a base, it can change the way the codons are read, completely change the amino acid sequence of the protein.
Wow, that sounds like it could really mess things up.
It can. Insertions and deletions can be really disruptive.
Are there any other types of mutations?
Yep. There are also chromosomal mutations. Those are changes to the structure or the number of entire chromosomes.
So not just single bases, but whole sections of chromosomes rearranging.
Exactly. Chromosomal mutations can involve things like inversions, translocations, duplications, and deletions of big chunks of DNA. Those can have really big effects on an organism's development and health.
Wow. We've covered so much. From the double helix to DNA replication, mutations, chromosome organization. It's incredible how much we've learned about DNA and how it runs the show.
It really is remarkable. And the story's not over yet. There's still so much more to learn.
I can imagine. What are some of the big questions scientists are still working on when it comes to DNA?
Well, we talked about DNA being the blueprint for life, but there's still a lot we don't get about how that blueprint is actually read and used. How do cells know which genes to express at the right time and in the right place? How does the environment affect gene expression? We're still figuring it out.
Those are some big questions.
They are. And there's also the question of DNA repair. Cells have these amazing ways to fix DNA damage, but we're still working out all the details.
It seems like the more we learn about DNA, the more we realize we don't know.
That's the best part of science. Every answer leads to more questions. It keeps us exploring, digging deeper into the mysteries of life.
Well, on that note, let's take a pause and continue this incredible journey into DNA in part 3. Okay, we're back, ready to wrap up our deep dive into DNA. It's funny, thinking about all those amazing experiments, a lot of what we know about DNA came from studying, like, bacteria and viruses, right?
You're right. They've been super helpful for scientists.
What makes them so good for studying something as complicated as DNA?
Well, for one, they're pretty easy to work with in the lab. They grow fast, too, which means they reproduce quickly. So scientists can look at lots of generations in a short time, see how mutations and genetic changes play out.
It's like watching evolution on fast forward.
Exactly. And their genomes are way smaller and simpler to analyze compared to, say, humans. If you're trying to solve a smaller puzzle is easier to figure out.
Makes sense. But can we really learn that much about human DNA from, like, a bacterium?
You might be surprised. A lot of the basics, like DNA replication, translation, the stuff we've been talking about, those processes are pretty much the same across all living things. What we learn from bacteria can often tell us a lot about more complex organisms, even humans.
So like a fundamental connection at the molecular level.
Exactly. It's kind of amazing, right? These little organisms have been so important in figuring out genetics.
Shows that even the tiniest creatures have a lot to teach us.
Absolutely.
Well, I think we've reached the end of our DNA journey. We've covered a ton, from the double helix to replication, mutation, chromosome organization, even those tiny but powerful bacteria and viruses, and how they helped us figure this stuff out.
Yeah, it's been quite the dive. And the craziest thing is, we're just getting started. There's still so much to learn about DNA.
It's mind-blowing. What mysteries do you think DNA still holds? What cool discoveries are waiting for us in the future?
DNA keeps surprising us, even after all these years of research. Figuring out its structure, that was just the beginning. How those DNA instructions are read and used, how the environment plays a role, even the nitty-gritty of DNA repair, it's all still being investigated.
There's so much to explore. What areas of DNA research seem most promising right now?
Oh man, genomics is just exploding right now.
Yeah.
DNA sequencing is getting better and cheaper all the time. Now we can look at whole genomes quickly. It's opening up crazy possibilities for personalized medicine. Imagine treatments tailored to your own genes.
So no more one-size-fits-all, but medicine made just for you.
Exactly. Imagine knowing your risk for certain diseases so you can prevent them. Or having drugs designed just for your genetic profile.
It sounds futuristic, but it's becoming real. What other DNA tech do you find exciting?
Gene editing. Stuff like CRISPR-Cas9, letting scientists make super precise changes to DNA. Imagine curing genetic diseases, engineering crops for better food, even making new biofuels.
We're on the edge of a genetic revolution.
We are. But with all this power comes responsibility, right? We've got to use these tools ethically and carefully. We need to talk about the implications of all this and make sure it's used to benefit everyone.
Absolutely. DNA is powerful stuff. We've got to use it wisely. Well, I think we've given our listeners a lot to ponder. From the basics of DNA to what it all means for medicine, agriculture, and the future of humanity, it's been a wild ride.
It has. I hope everyone listening is feeling inspired to keep learning and asking questions. The world of DNA is full of amazing things and possibilities. Who knows what we'll discover next?
Great point to end on. Thanks to all our listeners for joining us on this deep dive. We hope you enjoyed the journey and feel ready to keep exploring the world of DNA.
Keep those questions coming. Keep learning. Keep diving in. The future's in your DNA.